Good evening, Raj, and good morning, student, and also there's colleagues, sort of friends from ITB or also from outside ITB. I'd like to welcome Professor Ramaraj. So for the today topic, so I think I will give you a brief introduction again of yours, Raj.
Okay.
Although it is for the third time,
Yeah.
fourth time. So Professor Ramaraj is from the Nicholls State Universities. So he is also now is a visiting professor at the Faculty of Industrial Technology. As you know that today is the last lecture of Raj. Today's topic is the Cellulosic Ethanol and Microbial Lipid as biofuel. So that one is now is the last lecture for this semester. For the next for the coming semester, probably I will ask you again to have
Okay.
another classes.
Okay. All right.
Okay. Raj, I think the time is yours for.
Okay. Thanks, Chandra. Good morning, everyone. I'm happy to be back again. So as Chandra said, this is my last guest lecture for the semester. Today's topic is going to be on cellulosic ethanol and microbial lipids as biofuels. Can everybody see my slides okay? Thumbs up.
Yes.
All right. All right. So, so I'm going to talk about an overview of what is biofuels, why we need biofuels, even though oil price is kind of went cheap for a while. So why we need to go in this route. Also, I'm going to give you like socioeconomic factor involving biofuels, how it will improve rural economy. And then I'm going to, close up with some research data from my lab, okay? All right, if you look at some of the energy around the world, in India, for example, they use in rural area, dry cow manure as a fuel source for heating and cooking. And in Africa, you have this, woods mainly for cooking. And in the temperate country, oat was traditional biofuels. Oat was used for a lot of purposes, including, for mules, horses, and laborers. Oat fueled the farming for a long time in temperate region. Then we, moved to non-renewable energy source. We found coal, natural gas, petroleum, nuclear power. This is what we've been using now. Coal has been going down for a while now in the US at least, coal use. And oil is what is dominating 20th century, and also even now we depend on oil as an oil economy everywhere. So just to show how oil is a natural product. One time these were all living organisms, and when they were die and decomposing, and at the high temperature pressure, it become organic rich sediments, and which become oil. So this is what they've been extracting for the last almost 80, 90 years. This is what running the world, and we depend on mainly as transportation fuel. Okay, oil has some bad environmental issue because it can release CO2 and has greenhouse gas effect and global warming. So we not only use oil for transportation, we use for electricity and also heat and turbine propeller and also for many industrial use. So it's been running the world for a long time, use of oil. So the foundation of modern society, we call the lifeblood of modern civilization is, right now is because of the oil. 20th century we call the oil century. And it's a non-renewable resource. It took millions of years to form, and it is, the way we are using the oil is unsustainable. Not only it's polluting, but it is unsustainable because it's only a finite resource available. At some point, we're going to run out of oil. So we need to find alternate fuel source to keep our lifestyle. Otherwise, it's going to go down. We depend on some other energy source, not only oil. We need to find something for the future. So that's why we use this green technology. We mainly the alternate fuel source coming from renewable sources, not only wind, solar, but mainly for transportation sector, we still depend on liquid fuel. So we use this biofuel, mainly ethanol and biodiesel. So it's sustainable because we can produce crop, we can get this every year, it's renewable. We can reduce pollution, and the technology is innovation and create jobs. And it is viable technology, even though it's still a little expensive, but it is still viable. We can do this as an alternate energy source. So let's look at the energy demand. Energy demand, if you look at US and China, China is #1 for demand for energy in the US, and you can see Europe and Asian countries. So these are the major part of the world that consuming a lot of energy. And the energy, as I said, has been mostly dominated by petroleum, natural gas, coal. And in the future, we are hoping this petroleum is going to go down, and we're going to increase the biomass and renewable source of energy by the year 2040. So that's what we are hoping for, so we can reduce the greenhouse gas and benefit the environment as well. So the imbalance of energy is basically supply and demand. The demand for fuel is increasing because of urbanization. People are moving to city, migration. So if you look at horse and buggy stage to modern life and living, more people live in the cities nowadays. So there is more demand for energy use worldwide. So the solution is to this energy crisis is biofuels, as I said, for various reasons. It's sustainable, and also technology is almost there to commercialize this. So what are the various biofuels we can make? We can have liquid biofuel, gaseous biofuels. Some of the liquid ones are bioethanol, biobutanol, biodiesel. Gaseous one are methane, hydrogen, syngas, and we call hydrogen, which is hydrogen and methane together. So not only there's a demand for energy worldwide, other motivation is, as I said, is to prevent pollution or reduce pollution. So we can have 3 birds with 1 stone. So we're gonna take care of the excess crop residue, reduce pollution, and we can produce energy. So those are the motivation for producing biofuels. So we can take care of the environment by reducing the greenhouse gas effect. So this is some of the examples to show you the fogs in India and China. This is what it used to be in US and Europe in the 60s and 70s. When these economies were going through rapid development. Now, this is what we are seeing in winter months in India, the smog because of air pollution due to burning of fuels. So when you put ethanol as your fuel source, at least partially, like, so ethanol contains 35% oxygen, so it is oxygenated fuel, so it can burn better, so you can reduce emissions when you put ethanol in your gasoline. So in the US, all 100% of gas we buy has 10% ethanol blend. For this reason, because it's called oxygenated fuel, so it burns better and reduces emission at the tailpipe. So what is ethanol? So ethanol is this molecule of C2H5OH, and this is how the structure of ethanol look like. and it has oxygen, so it burns better. So it has less, pollution. So you can make ethanol. You need sugar for it, and the organism that does that most commercially is yeast. Yeast can ferment glucose and make ethanol and CO2. From one molecule of glucose, yeast can produce 2 molecules of ethanol and 2 molecules of CO2. So this is what it is structurally given. Here's your glucose is your raw material for this, and yeast is your biocatalyst, and you produce pyruvate, and then you make ethanol. Okay. So in a little more detail. So the first step in glucose conversion to ethanol is glycolysis. So during glycolysis, glucose is converted to pyruvate. And then this 2 pyruvate through acetaldehyde, it's going to produce ethanol, and you can regenerate the electron carrier NAD back and forth carrying the electrons for this whole biochemical reaction. So ethanol generally commonly known as grain alcohol, mainly because it's most of the ethanol come from corn in the US and also other grains and potatoes. These all contain starch. Starch is made up of 100% glucose. So when you break the starch, you get glucose and you can produce ethanol. In Brazil, they use sugarcane as a main raw material for ethanol production. So it is energy efficient, making ethanol from corn. It yields 25% more energy than we use energy to grow the corn. So it is energy efficient. If you look at energy output input ratio, it's 1.6 ratio. So by doing this way, if you wanted to produce corn for only fuel purpose, it is still energy efficient. So if you compare gasoline with ethanol, and you can see this octane number, it's comparable. The main fuel source, of course, gasoline is crude oil. Then you can make a variety of sources for ethanol, from corn to agriculture waste to grain to sugarcane. Energy content is equal, almost, quite good. Gasoline is 109,000 to 125,000 BTU, whereas E85 ethanol is 80,000 BTU. Energy ratio is compared to gasoline is 70%, and both are liquid fuel. So this is what is needed currently for transportation sector. So if you take starch, mainly from corn and potato or anything else, which is made up of 100% starch, you need to hydrolyze starch by adding amylase enzyme, and you get glucose. And this is your raw material for ethanol. As I said before, it undergoes glycolysis via pyruvate and acetaldehyde, and you make ethanol biochemically. So first you need to add variety of enzymes. So here you need amylase to break down starch to glucose. This is how it is done commercially. And then you need pyruvate peroxidase, which is basically the yeast can produce these enzymes, oxidoreductase, acetaldehyde, hydrogenase, and ethanol dehydrogenase. So these are all produced by yeast itself, but here we are adding amylase to break down starch to make glucose, and from glucose onwards, yeast will take over and produce ethanol. So yeast is this amazing biocatalyst. It can produce ethanol variety of ways. Most yeast use 6-carbon sugar, and some of them yeast can use 5-carbon sugar. So we have pentose and hexose sugar. So the biochemical pathway is glycolysis and pentose phosphate pathway. The organisms can do fermentation reaction and variety of way to make your ethanol. And also it assimilates some of the carbon for growth. And so that's why during glycolysis, it gets 2 ATP, so it can use this energy for growth. Again, that's your biochemistry of starch fermentation. And again, from glucose to ethanol, yeast can produce these enzymes to take over. So you need to make glucose from starch. So this process is done adding amylase in ethanol factory. So the steps involved in corn ethanol is you take the raw material that's corn or wheat, go through milling, and then we do this enzymatic hydrolysis. This is where you add your amylase to break down starch to glucose, and then you go through mashing, and then you add your yeast. Fermentation reaction takes place in big tanks, and then the product separation takes place, and then you make ethanol. And dry distillate grind is one of the byproduct, which is used as animal feed. Has a lot of protein in it. Okay. So that's steps involved in grind ethanol. So milling, liquefaction, saccharification, fermentation, distillation, dehydration, denaturing, and co-products. So those are various steps involved in making corn ethanol. Okay. So currently, ethanol from corn industry is a mature technology, and we are making commercially a lot of ethanol from corn. And if you look at this map in the US, we have more than 200 bioethanol plants, and they make 10 billion, close to 10 billion gallons of ethanol every year. So we are making ethanol enough ethanol to make 10% additive in our gasoline. So this is commercially mature technology currently. So how do you make ethanol from cellulosic material? We are talking about agriculture residue. We're talking about wood chips and all the biomass. How do you make that? So cellulosic ethanol is mainly made from cellulose and also xylosic sugar. Cellulose main purpose is to provide structure to the plants, and you can get this from any plant material that is currently we are burning in agriculture crop in India and China. So you can some way to economically harvest after you produce your food crop. You can process this agriculture biomass and get sugar out of this cellulose and hemicellulose. And then you can use that as raw material to make the same process like I showed you for corn ethanol to make ethanol. So the process concept is simple. You take the agriculture residue and do chemical treatment to delignify, remove the lignin, get cellulose available, xylose available, and then you do enzymatic hydrolysis to get your glucose and do fermentation and you make ethanol. That's a simple concept of how this technology works. So there are a lot of biofuel crops in the market. People currently use mostly sugarcane and some agricultural residue from wheat and corn plant, maize. And if you look at it, this is a power density of each of those biofuel crops, okay? So the sugarcane, we like it because it's a high power density. So the main purpose of the cellulose, hemicellulose, and lignin is to provide plant its structure, okay? So plants make this to make it structurally viable molecule, so your plant can withstand a lot of those pathogens and protect the plant. So it's basically made up of glucose. If you look at cellulose, it's 100% made up of glucose. And then we have hemicellulose made up of some pentose and hexoses. And then lignin is made up of polyphenolic compound. So here is, when you break it down, there's three major components of the plant material. They are lignin, cellulose, and hemicellulose. And this is your hemicellulose, and this is your cellulose. And when you break down lignin, lignin is made up of this polyphenolic compound. These are the major one. Fumaryl alcohol, coniferyl alcohol, sinacol alcohol are the major components of this lignin, which is polyphenol. So just to show you what are the different sugars in hemicellulose, we have this, the hexose and pentose, they're made up of xylose, mannose, glucose, galactose. We combinedly call them xylosic sugar, which makes this hemicellulosic component of the plant. And if you look at cellulose, it's 100% made up of glucose. So if you can make some kind of economically viable pretreatment to break this and release cellulose and hydrolyze cellulose, you get 100% glucose. And this is going to be your raw material for your ethanol fermentation. And you can also use this sugar as well. And we have this phenolic compound. This phenolic compound could be used to make biodiesel, okay? So the composition in general, it varies from crop to crop. 15 to 25% of plant has lignin content, hemicellulose 23 to 32%, and cellulose 38 to 50%. But it varies. Some plants have more cellulose, and some plants have less cellulose. So we can use grass, you can use any plant material to make ethanol. So if you look at any plant that takes the solar energy and make this fiber, and this fiber can be harvested and pre-treated to release glucose, and then you go through the fermentation and make your ethanol. And we can use this ethanol for transportation purpose. When you burn your fuel, you're releasing CO2, which can go back, and the plant can take the CO2 and fix the CO2, make the biomass. So it is kind of a carbon neutral. You're closing the loop. The carbon cycle is complete if you use this biofuel technology. Is this environmentally sound? As I said, it is environmentally sound, but the cost prohibition of making this to work is to get this fiber, you harvest, and it is voluminous. You need to kind of, transport them to central locale and do pretreatment and get the sugar out and go through your ethanol fermentation process. And then whatever waste comes out, you can use as cogen to make your electricity. So it is environmentally sound, but again, we're competing with cheap gasoline. So that's where the debate comes into play, how to make it viable technology. So once you get your material, you have two possibilities. You can go through biochemical route or thermochemical route. So here are your feedstock. We are talking about agriculture residue. You can use forest residue, or you can grow energy crops that are mainly grown for doing this energy production. And then now we can use municipal solid waste as well for this purpose. So you can use hydrolysis, fermentation, biocatalysis for biochemical route. Thermochemical route, you can use combustion, gasification, pyrolysis. And in this route, you can make variety of product. And here you can make electricity, heat, steam, and plastics and other material, bio oil, and all that stuff you can make. So you can use, if you look at the landfill, what is in the landfill, 70% what's going in the landfill is lignocellulose material. So if somehow we can separate and get this, that could be additional raw material for your biofuels, okay? In the US, we have variety of biomass available. We have energy crop. People grow energy crop for this purpose, especially sugar cane. We call it energy cane. And we have agriculture crop residue from corn, wheat, rice, cotton, whatever left on the field, okay? 10% go back as a fertilizer as a soil amendment. 90% of the crop residue could be potentially used for lignocellulosic material. And then we have forest residue from sawmill where we produce our lumber. As I said, landfill is a potential source of lignocellulosic materials. So based on all that, the USDA come up with an estimate of 50 billion gallons of waste. From the waste, we can make ethanol. USDA come up with an estimate of 80 billion gallon possibility. 1/3 could be used in transportation industry. By 2050, we can produce this energy crop switchgrass on 114 million acres. That's a possibility of making 165 billion gallon of ethanol per year. Currently, we're making roughly 10 billion gallon of ethanol. So there is a potential possibility to increase tenfold of ethanol from energy crop and variety of agriculture residue. Again, so you need to grow some energy crop, or you can use the waste material, bioprocess it, go through a series of pretreatment and go through fermentation. Once you get the sugar, then the step is the same like corn ethanol, which is a mature technology. And then you can use biofuel for whatever purpose you can use. But you still produce some emissions. But as I said before, comparatively speaking, it's kind of carbon neutral. Almost all the carbon we generate can be recycled back. When we grow plant again, plant fixes the CO2. So this is what we call now new crude, or the crude come from the agriculture is our new crude for our biorefinery. So once you process the various raw material, you can make this C5 and C6 sugar, and you can treat this sugar from agriculture sector as your oil. So exact concept of refinery. We call this biorefinery, so you can make variety of product. Talking about ethanol, diesel, jet fuel, most of the material. And also you can use the sugar platform and make fine chemicals. So depending on the need, once you get the sugar, you can produce a variety of products. So there's a lot of advantage, as I said before. It benefits environment, it benefits agriculture industry, and there is food security because we are mainly using base material from agriculture crop. And the main advantage is less greenhouse gas, reduced dependence on oil. Most countries import oil, so we can have almost energy independent, okay? And diversify your agriculture economy in rural side of the country, okay? So once you get your raw material, you can make a lot of fine chemicals, you can make energy, whatever the need of the country, you can make. The technology is there. The challenge is to get the sugar, and once you get the sugar, you can use the sugar platform as a biorefinery. So we started out with first gen, which is our corn and sugar to make ethanol. The second generation is the lignocellulosic material. The third generation is biofuel from algae. And the fourth generation is biohydrogen. So right now we are talking about these two technology. So we're talking about biofuel making biodiesel, and also talking about making ethanol from agriculture waste. So you can make a variety of raw material from energy crop to agriculture residue. We can use them all, but you have to process them in a way, and it should be economically viable technology. So main thing is if you look at the structure of the plant, all these are bound together, so it's not bioavailable. So it's a complex structure. So you need to first remove lignin and get the cellulose, hemicellulose out of the plant fiber. And then you do pretreatment to get the sugar out. So there is a lot of process involved to get the sugar out. So first you need to remove the lignin, and then you get the sugar out of these polymeric structure. So this is how it looks like. So you need to somehow separate lignin out of the plant fiber and release cellulose, hemicellulose, and then you enzymatically hydrolyze them and get the sugar. And then you, once you get the sugar, the technology is same as corn ethanol. All right, the motivation started here because of when oil price went up to $100, almost $100 a barrel, and President Bush at the time promoted biofuel as a way to go. And that's how the technology started really picked up really fast in the US. And so again, the motivation is the price of gasoline went up in the early part of 2000. So I want to emphasize some basic fact about what we really need. We what we really need is the services that energy provides. The service we get from energy is heat, mobility, light. Those are the services we get. Energy has fundamentally different qualities, and all carriers are interchangeable, and all BTU are not created equal. Industry, the society literally stops if you don't have any liquid fuel. So the liquid fuel is the key for our society now. So we need liquid fuel at least for next few decades until, you know, the solar energy and until we get the battery powered car has become norm of everyday life. So this is just to show you, if you don't have liquid fuel, everything will completely stop in the world currently. So let's talk about all the energies. We talk about coal. We have plenty of coal, no problem. Natural gas, there's a lot of natural gas currently in the US. We import from Canada and Mexico. Because of the fracking technology, we produce more oil nowadays in the US. As I said, it's not sustainable. At some point, we're going to run out of oil, so we need to depend on finding new technology, especially biofuels. Okay, so also petroleum dependence undermines a lot of things: climate security, economic security, international security, and world stability. So for these reasons, we need to get away from petroleum and try to go into this route of biofuels. So biofuels, including cellulosic ethanol, are alternative that can provide all this, as I said, greenhouse gas reduction, economic advantage, national security. And the rest of the talk, I'm going to focus on from agriculture residue, how to make ethanol. Okay. So let's go through the process. What is commercially now successful? So from corn, this technology is mature. We make 10 billion gallon of ethanol. So you take corn, get the hydrolytic process, get sugar, fermentation, distillation, drying, you make ethanol. This is well-proven technology commercially in the US and in Brazil. We make plenty of through corn. This is from sugarcane, from Brazil. Instead of corn, they use sugarcane. Okay. If you look at the cellulosic biofuel, so from you get the plant fiber, pre-treat them, do the hydrolysis, get the sugar, and from once you get to this point, everything is the same. So here you need to work on reducing the cost, and once you get the sugar, and the technology is the same. And then the other possibility is you can take this plant fiber and go through your thermochemical route, and you can produce heat and power and fuel and chemicals. So these are a variety of possibility that we can do, whichever is suited for a particular country. So let's look at major cost elements. So if you look at one point, when gasoline was produced first, the raw material cost was high. Bioprocessing cost was also high. As a result, the oil cost was higher when the technology started. So you have cost of raw material and cost of processing, right? So there's two costs involved. So if you look at the cost to make them, 70% petroleum costs and processing cost 30% currently. But at the beginning, getting this was also high, okay? Because of this R&D science, we reduce the processing costs constantly. So the same thing applied for biofuel. So we have this industry depends on the cost of oil. We have this margin of oil price to work with. So here is the cost of your biomass. So we need to reduce the cost of producing ethanol from biofuel, the processing cost. So we have, as long as your barrel of oil is 60, 65, it is still possible. we can make this economically viable. Okay, if the price of oil goes to below, then our process cannot compete. So there is a big margin to play with it. So that's why the R&D is key to reduce the processing cost of this technology. So I'm going to show you some example how this all first started for gasoline industry. When gasoline was produced first, early years, you have this cost of getting the oil out, but the processing cost was high because this technology was improving. People are making, how to cheaply make various products from crude oil, right? And now if you look at it, it flipped. The processing cost is low because of oil price going up, the raw material cost is high, right? In the future, the raw material cost is going to go up because we're going to use more oil, there's no more oil to get. So this price is going to go up. So your processing costs can reduce a little bit more. So the same scenario is going to play for cellulosic ethanol, okay? So we're going to look at ethanol production from Brazil. And this slide I borrowed from Brazilian colleague. And you can see that the ethanol production cost went down in Brazil because of R&D, because of science input. They reduced the cost, okay? It's comparable to gasoline cost. So the same thing you can, like oil, how at the early stage, they went through this, we can do the same thing here. So here is the biomass cost today. The processing cost is high. What through science they're trying to achieve is reduce this cost. So you're going to have overall economically viable energy coming from biomass. So we want, but through our constant evolution of science and technology, we are trying to reduce the cost of processing the biomass. And so we can have overall a cheaper energy source. So that's, so we're doing this by learning by doing in large scale because a lot of people are working on in a lot of different labs. So in cooperation, so we can reduce this cost and in the future, we can have comparable energy to oil. And it is attractive mainly because any country can produce this energy. You don't have to depend on oil producing country for your economy. It improves the environment. And again, it's going to improve a rural economy because you produce these crops in rural side of your country. So in the US, currently we have, these are the plant that commercially put up, producing various raw material from corn stover to vegetative waste. This is a capacity of each plant that produce ethanol, okay? So most of them use this AFEX pretreatment technology, which is the ammonia fiber expansion for pretreatment. I'm going to show you what that means in a minute. So if you look at the whole process is you get the crop, take it to storage, reduce the size because these are bulk and voluminous, and then you pre-treat them. The pre-treatment is done to remove the lignin, right? And this is where there's a lot of different technology out there. AFEX is one of them. And then you enzymatically hydrolyze and get the sugar out and go through ethanol recovery and residue utilization waste treatment, okay? And you can also hydrolyze the conditioning and go through the hydrolysis side for fermentation and go through ethanol recovery. So here we are talking about this part to reduce the cost of production of ethanol from cellulosic material, okay? So that's what the whole design is. So we're working on this part. And from here to make ethanol, this technology is already matured from corn industry. The process is the same. You're trying to get the sugar, trying to get this pretreatment cost is what people are working on reducing it. So this is how it looks like overall cellulosic biomass to produce ethanol. You get the raw material from different sources, go through pretreatment, solid liquid separation, enzymatic hydrolysis, fermentation, product recovery, and then you can do the hemicellulosic part and use the hemicellulosic sugar and go through making ethanol, residue processing, and co-product use. Okay. So let's talk about one of the commonly used pretreatment method, which is called ammonium fiber expansion. In short, it's called AFEX. AFEX is basically you take the biomass, put in a reactor, and you put ammonia and heat, and so it expands, and you get the lignin out, and then you can recover the ammonia and reuse the ammonia, so you can have reduced cost. And the treated biomass has cellulose and hemicellulose, which can be processed further. So rapid pressures release ENS treatment, and 99% ammonia is recovered and reused. The remaining serves as a nitrogen source for downstream for fermentation. So AFEX process is commercially successful in the US. And of course, it's a patent technology. So anybody that uses, you have to pay royalty. This technology, sugars are not degraded. You get 100% sugar available for fermentation. No inhibitors are produced. Okay. So based on AFEX modeling, and the cost of producing ethanol from crop residue is $1.41 per gallon from AFEX technology, compared to acid pretreatment, hot water pretreatment, lime and corn dry mill. So this technology is right there, and they're trying to reduce the cost. Currently, that's what the cost based on what is the ammonia use and the technology use. So result of economic analysis show that you can reduce ammonia loading. People are constantly tinkering with that. You can reduce the required ammonia recycle concentration and reduce capital costs. So people are working on it, and then they're saying that in the future, if to process 2,205 dry ton per day scale, you can make ethanol for 62 cents a gallon. I mean, this is what they're predicting, right? If this happens, I mean, this can be a lot cheaper than gasoline price currently. So this is based on a lot of modeling, and this is possible using AFEX in the US. Okay, that this is. So when it matures, this technology, when a lot of people use this will even go down to below 60 cents a gallon. So because of inexpensive ethanol, so environment improvement is possible, petroleum dominance going to decline, and rural economic development is possible. So these are all advantage of using ethanol from biomass, especially agricultural waste, okay? And again, it is not competing with food. So there is no food versus fuel debate. We are talking about crop residue. We are not competing, making crop for energy purpose. Okay. So based on all this modeling, the Michigan State University come up with this technology, how it can be, transferred. So you get, you need to have all this pretreatment industry in the rural area in a cooperative way. So you don't want to have one big mill. You want to have a lot of small mills all around different farms. So say 150 farmers can get together and make a cooperative regional bioprocessing center, right? Biomass processing center. And then we can collect raw material, whatever raw materials produced at this particular area, you can process them in different ways. You can produce high value use like protein enzyme, power plant, biorefinery, make ethanol, and treated biomass go back to agriculture use. So this is what they're predicting the way of the future. So make a lot of small cooperative biomass processing center in rural economy. And this is supposed to help a rural economy in the US as well as in other countries. So let's talk about some of the myths out there. So if I talk to my friends around here, they even have the same question. So they say ethanol has negative net energy. People say that. Say gasoline's net energy is worse than ethanol in reality, okay? Ethanol will drive up food prices. People say that. It's complicated. It's no easy soundbite for that. Cellulosic ethanol will reduce the food prices because we are working on using crop residue. Some people say ethanol is bad for the environment compared to what? Corn ethanol is actually superior to gasoline now in most metrics if you look at it. So cellulosic ethanol is even better, okay? Ethanol will always cost more than gasoline, but if you look at some of those modeling people are working on, especially in NRA, which is in the US, a major research lab, they're predicting it's getting cheaper in using modern technology. So we need to have a way to get the lignin out. Afex is one of the successful pretreatment, but there are a lot of other pretreatment out there. But the way I wanted to focus on using biological process, so you can use some fungus to get rid of this lignin instead of going through even Afex or acid or alkaline pretreatment. So if you look at the steps involved, you need to do the pretreatment. And then you get the sugar out, you get ethanol. So this is where Afex is only successfully proven here, but you can work with other pretreatment method. My focus is work on biological pretreatment using fungal enzymes. So we have different pretreatment. We have acid hydrolysis, alkaline hydrolysis. Here is your ammonium fiber expansion. I showed you how that works. A lot of people use ionic liquid pretreatment, right? This is still expensive, it's not commercially viable yet. And so far in the US, this is the technology that's been proven commercially viable technology. So the focus for me is to use microorganism itself, right? And especially fungus to use this peroxidase enzyme so you can get the lignin out in one big reactor. So that's what I'm working on. Okay. So enzymatic biomass treatment is very helpful. You can reduce the cost. You can reduce the water use. Enzyme can be easily recovered and reused, and you don't have to have complicated technology for that. All you need to use the right organism to produce the enzyme. So here's just to show you the water requirement for various pretreatment. And so it's really high. All the red bars for different processes, the water use is really high. But if you use an enzymatic technology for removing lignin, the water use is less. So that's another advantage of using biological enzyme to remove lignin. So I'm focusing on using lignin enzyme, peroxidase enzyme. It's a multi-copper enzyme belonging to a group of blue copper protein. Lacase are mainly produced by fungi. And we are working with the white rot and brown rot fungi to remove lignin. And it really works well. It's not very complicated. It's a well-proven enzyme working for various purposes like degrading dyes and various activity you can do with this enzyme. So these enzymes are produced by a lot of different organisms, like especially insects like termites. Inside there's microorganisms that can process the wood these termites eat, and they can produce a lot of variety of enzymes. So the same function is enzyme. So you want to use the enzyme to remove the lignin, and get the cellulose and hemicellulose out and go through hydrolytic steps. Okay. So that's how this technology works. Instead of using other pretreatment method, we are looking at using lignin peroxidase enzyme from white rot and brown rot fungi, and get the molecule out and go through saccharification step and get your ethanol in this way. So that's another way to do that. So when you use this enzyme, so it breaks this lignin. You can see that in electron microscope before and after treatment, and delignified cell, and then you can get the cellulose and hemicellulose out of this complex matrix. And that's how it looks like after you treat them with the fungal enzyme. You get the pre-treated biomass. So I'm going to show you some technology, how we are going to use the fungal enzyme instead of using FX or acid treatment or alkaline pretreatment. We can use fungal enzyme to produce your raw material for fermentation. So this is my research that in my lab, we did that some time back. I'm going to share the result. Again, this introductory slide is the same thing I just went through. We need a liquid fuel. So we are trying to get it through biomass, and this is carbon neutral. Currently, we are not producing enough. We need to produce more biofuels. Okay, if you look at it, 1% of the world energy demand is by biofuels. So the U.S. government set a lot of goals. They keep changing that now, depending on the gasoline price. Now it went to $1.50 gallon for ethanol. It used to be $2.33 when gasoline price was high. So we work with the sugar cane, especially the energy cane, because in the state I live in, it's a sugar cane state. They have a variety of this cane that it has high biomass and less sugar. But the advantage of using this energy cane is this can tolerate cold weather because this sugar cane in Louisiana, they harvest in September because before freeze comes in. But this energy cane can withstand cold, so you can grow them all through the year, and also you can grow them in less agriculturally viable land, marginal land. It grows really well. That's the reason that the state is promoting this for energy production. So if you compare the energy cane with regular sugar cane, here is the energy cane. It has more biomass. If you look at it, the fiber content is more compared to the real sugar cane that produce sugar. So we want to grow this sugar cane and get this high rich fiber and process this fiber for producing ethanol. So we started out, collecting the sugarcane from this USDA sugarcane lab and processed it in the lab. And again, we need to remove the lignin and get the cellulose, hemicellulose out. Once you get the cellulose, it's 100% glucose. And then once you get the hemicellulose, you get pentose and hexoses, and then lignin is phenol. So we want to use all three polymeric structure in this plant fiber. Okay, I'm going to show you how we did that. So you just take your raw material, in this case sugarcane, pre-treat them. I'm going to show you some acid alkali pretreatment, and then I'm going to show you how we use the fungal enzyme, and then we did the fermentation distillation. So this is some of the objective of our research, why we did that. And we first started with acid pretreatment. We want to compare the result with acid, alkaline, and our fungal enzyme. So here is our acid pretreatment, dilute sulfuric acid we use. And then there's a fungal enzyme we process from this white rot and brown rot fungus. So basically you grow this fungus right on the crop itself, the cut biomass itself, put in a bag, spray this fungus, let it grow and soften the lignin. That's what we did. And then we went through enzymatic saccharification. Once the fungus was treated for a couple of days, we take the biomass and go through enzymatic hydrolysis to get your cellulose and hemicellulose. And we used first yeast, and then we used our own recombinant E. coli that can process both sugar, hemicellulosic sugar and cellulosic sugar. So we worked with yeast as well as these recombinant E. coli. So here's the result. So here's the acid pretreatment, different 1% to 4%, and you see ethanol yield, not much. This is without enzymatic hydrolysis, just acid pretreatment, whatever sugar released, okay? So you can see that the more acid you had, the more ethanol we produce. 4% and 3%, there is statistically no difference. 3% acid seems to be the optimum acid pretreatment for this, okay? And this is alkaline pretreatment, just to increase the pH with sodium hydroxide. And if you look at the pH 13 and 12, there is no statistically any difference in yield. So pH 12 worked as well as pH 13. And here is the fungal enzyme. So here is a white rod and brown rod, and you put them all together. So when you spray with biomass with both Cereoporiopsis and pheneric, you produce more ethanol compared to individually you treat them with this fungus, okay? And just to show you what amount of glucose we produce in all this, here is your alkaline treatment, here's acid treatment, is a fungal treatment. So we produce statistically significant amount of sugar in all these 3 different pretreatment methods, your glucose yield, your xylose yield, okay? So now we want to combine this fungal pretreatment with sulfuric acid pretreatment, so you can reduce the acid use in your pretreatment to make it commercially viable. The idea here is when you harvest the sugarcane, you're going to store it. When you store it, the biomass, before you process it, you spray it with this fungus for a few days. As a result, you can reduce your acid. Instead of 3%, you can use the acid to 1%. So that's going to reduce your pretreatment costs. So here we've sprayed for 2 days of fungus, and then we treated them with different acids. And you can see this 1% acid produced as much as 3% without fungal treatment. So we can reduce 2% of acid use by using additional fungal treatment during storage of this biomass. That's what we're trying to do here. We can reduce this cost of pretreatment by using biological process. That I showed you is without enzymatic hydrolysis. Now we work with this E. coli, which is, we did this work to get the E. coli that can produce sugar from both cellulosic and xylosic sugar. We put some genes in this to work on it, just to show you how we did it. We did some knockout experiment, and we used this Zymomonas mobilis alcohol dehydrogenase pyruvate carboxylase into E. coli. We mixed acids, we knocked out and produced only ethanol, okay? So just to show you, we knocked out these genes. So the bacteria are not producing any of this product. It only produces ethanol. And in this way, you can make a 95% yield of ethanol, okay? And just to show this concept, this is the xylose and ethanol. We grow them in the lab in a 5-liter fermenter. You can see the xylosic sugar go down because we put the gene in this bacteria. And then the biomass, bacterial biomass going up, organic acid going down. When we did the knockout of these acids, you increase the yield of ethanol. You don't produce any acid here after you knock out the gene. So we improve the yield, we make a theoretical yield of ethanol from almost theoretical yield from using this genetically modified bacteria. We just proved a point. We did the pilot scale using bagasse, which is one of the solid waste come out of the sugar factory. And we used a lot of this enzyme to get the sugar out. And then fermentation was done in a 5-liter fermenter. And you can see that we are producing, reducing all this sugar, total sugar, glucose, xylose come from this processed, pre-treated energy crop, bacterial cell mass going up, and ethanol yield going up. So we have proven this technology that you can combine biological pretreatment with acid pretreatment, reduce the use of acid, you can reduce the pretreatment. You can make ethanol from your biomass, okay? So this is the one research we did in the past, and just want to share the result. Now I'm going to show you, we have a lignin left, right? The lignin has a lot of phenolic compound. I want to show you how the phenolic compound can be used to produce microbial lipids, and the microbial lipids can be used as biodiesel. And the rest of the time I have, I'm going to share that. So again, the need for this is why we do microbial lipids, the same need, because we need a transportation fuel. That's what currently the energy is mostly used for transportation. That's why we are finding different biofuels. We have US mandated to make how much biofuel. So we are not able to meet that standard. We're trying to still way below. We are only making 10 billion gallons. And by 2022, we are supposed to be making more than 35, close to 36 billion gallon of renewable fuel. We are not able to meet that yet. So we need to find a lot of different ways to meet the US government mandate. So let's talk about biodiesel. So biodiesel nowadays produced from jatropha and other oil seed. One of the problem is it produces methyl bromide. Methyl bromide, is one of those bad things in the environment. The European Union banned that. You don't want to produce this. So this is one of the byproducts from oil seed, okay? Another problem is palm oil is a poor biodiesel because it congeals at low temperature. It has to be catalytically, you need to crack it or mix it. It also it needs a shorter chain length. So you can do that by biologically putting antisense elongases in the plant. You need more mono unsaturation, so you need to engineer desaturases. So there are a lot of things need to be improved on in palm oil. So you can do genetic engineering to do that, modify the genes in the plant. So once you make the oil, you can go through processing and you make your biodiesel. So only by bringing genes from elsewhere you can do that. So you put those genes in the plants, so you can make your biodiesel from plant work better. So you can reduce those poor quality you produce currently. So what are the lipids? Looking at wax, which are polymers or long fatty acid, cutin, polymers or short fatty acids, and then we have triglyceride. These are the 3 fatty acid chain bound to single molecule of glycerol. So these are all the lipids we're talking about. Biodiesel come from various sources. We have, mainly parent oil source. You have this triglyceride, and then you add methanol and catalyst, and you produce glycerol, and you make your biodiesel methyl esters. So you are familiar with jatropha for biodiesel? Jatropha, Indonesia, Malaysia, they produce a lot. 30% of seeds get, $140 per ton, optimistically. Fruits, you hand harvest, dry, and seeds are removed by hand. We really don't know whether it is economically viable or not. So some more information on Jatropha. It's a common name is black walnut nut, purge nut. Common oil name is hell oil. It produces this toxin. That's another problem. When you process it, you get this cursine, similar to ricin from another plant that I'm going to show you. So there's no antidote for this poison. Jatropha poisoning is it resembles organophosphate intoxication. So what to do with this toxic byproduct is another problem of Jatropha. As I said before, this is some of those yields of Jatropha variety. How much cursine is produced when you process them is very almost equal to ricin, potent toxin. Cursine is heat degradable. Okay, so castor is another. You can produce oil. Castor, a similar problem like Jatropha, and here you produce the toxin ricin, right? So from 1 milligram per kilogram of toxic, you produce 50 liters of 13 gallons of thing. But the ricin byproduct can kill 3 people. So there is some problem of this byproduct from Jatropha as well as ricin. So that's the reason that in some other state in US, they banned processing of these seeds. So you cannot, process castor in Oklahoma because of this production of ricin and curtine. So that's the problem. In some other states in the US, they banned. So what is the solution for making biodiesel? So you can go to algae or you can go to microorganism like bacteria, right? I'm going to show you how you can make from bacteria, right? So that's another source of making biodiesel. So people are working on algae to make biodiesel. They make methyl esters like exactly like biodiesel product. I'm going to show you how to use this lignin polyphenolic compound that come from the sugarcane processing. You can make biodiesel. Okay. So as I said before, the oil price is cheap nowadays. As a result, some of these companies I showed you at the beginning of the slide went bankrupt because they couldn't compete with the oil price. And now the oil price is stabilizing a little better, but when the oil price went down, this company has to shut down their plants that produced lignocellulosic ethanol. So what is biodiesel? As I said, it's a transesterification of renewable oil, non-toxic, biodegradable. You can get less greenhouse gas emissions. Most common feedstock, soybean, algae, waste animal fat, and oil. But the feedstock cost is high. If you take from these sources, it competes with food source, right? So you take methanol and add catalyst and do the transesterification and you make biodiesel, and byproduct is glycerin you produce. So we are using oligogenous microbe in this case, Rhodococcus opacus. And this organism accumulate more than 20% of biomass as lipid, okay? It stores these lipids intracellularly as a reserve. That's the natural part of why it is storing. It is water-soluble triester of glycerol and fatty acids. It happened during the stationary phase of the bacterial growth. It occurs in the presence of excess carbon and nitrogen limiting condition. When nitrogen is limited, the bacteria makes this oil as a storage material, lipid inclusion bodies. So we are exploiting this naturally occurring phenomenon. So we're using Rhodococcus. Why we are using that is an alternative to animal and plant oil. So you can control this process when you use microorganisms. And it's not impacted by season and other problems when you're dealing with plants. Lipid extraction is some technology needs to be worked on still. We are still working on cost-effective carbon source and identifying more oligogenous microorganisms to process the waste to oil. So some of the economics of it, if you start from soybean, if you start from miscanthus, when you use bacteria to make oil is more efficient than from plant source, okay? So if you work out economics, 9 times more oil than soybeans and miscanthus. So microbial lipid is very, very viable technology. So the goal is to take waste, in this case, the lignin processed after your ethanol production, the lignin phenol is still there. We use this as a carbon source, grow rhodococcus, and allow the bacteria to make oil, transesterify, make biodiesel, right? So that's the goal of this project. So we use the rhodococcus, rhodocrus organism, which is a gram-positive red pigmented, used in mostly bioremediation industry because this organism can degrade hydrocarbons and PCBs, polychlorinated biphenyl. So this is the ATCC culture. It belongs to actinobacteria, which include all these organisms, Nocardia, Gordenia, and Rhodococcus. There is limited knowledge of liquid accumulation by this bacteria from the waste product. We know it is making from glucose, but we want to see whether you can use this from as a bioremediation organism and also from waste product to make lipid. So we started out first to show the organism can make lipids from glucose. So this we use sugar first, and just to show you how much lipid you can make. And so we started out with glucose just to show you glucose goes down, lipid accumulating in bacteria. These are different concentration of sugar in the culture medium. And this is how the bacteria look like after we grow them in nitrogen limiting conditions. You can see the lipid inclusion in these organisms, okay? And then we looked at what type of fat in these lipids. So we processed the lipids and we found them, it's equivalent to biodiesel. So most of them are C16, C18 methyl esters, and it's equivalent to biodiesel in property. Okay? And just compare just glucose used by other organisms. The organism we are using is comparable, and 3 to 5 days you make this oil in your reactor. This is just the glucose compared to what is in the literature using other organisms. So it's comparatively good organisms to use. And now we are going to show you the processed waste from this sugarcane, especially lignin, how we can make microbial lipids from the lignin. So again, when you process the biomass, you're going to get this component, and glucose, you can make some inhibitors when you do pretreatment, and xylosic sugar you can use for making ethanol. And now we are going to look at what happens to phenolic compounds. So these are the waste material. We are trying to use this waste material to make microbial lipids. So first we show whether the bacteria can grow on xylose. Yes, it can, but it can, if you combine with glucose, it grows better. And you can see the cell dry weight, and you can see the concentration of the sugar going down, and you can see the percent lipid accumulating. Okay, so yes, you can use xylose as well for this purpose. And again, to show you, it produces the same quality of FAME profile, and it shows biodiesel property by using xylosic sugar, okay? So just to show how much oil, you can see this, the color of it. There is some inhibitor come from when you do pretreatment. We can also use this inhibitor. The bacteria can break down this inhibitor and make biodiesel. So we are seeing this organism is versatile. You can use glucose, xylose, inhibitors as a raw material to make biodiesel. So let's look at some of the phenolic compounds. So when you look at phenols, a lot of different phenolic compounds come out. So we are showing that phenol could be used by this organism to produce microbial lipid, but it needs little glucose at the beginning. You combine with glucose, it does better, glucose plus phenol. Phenol alone at the beginning, it didn't work, okay? So when you combine with glucose and phenol, it works, but phenol alone, but after several exposure to phenol, it started to work, okay? And again, just to show me same profile from phenol, it's produced same palmitic and oleic acid, same property. There's no difference whether the raw material is glucose or phenol. It produced the same thing. So we did some basic research to show you what's going on, what kind of genes are turned on, what kind of enzyme is produced. Without going through much detail, when phenols are broken down, it produces acetyl-CoA. The acetyl-CoA is going through fatty acid synthesis to make methyl ester. So this is what is going on in the organism to break down phenol by regular phenolic metabolism. And the end product is acetyl-CoA, which is taken to the fatty acid synthesis pathway. Okay? And just to show you, when you use phenol, which genes are turned on, when you use glucose, which genes are turned on and turned off, just to show up regulation, down regulation based on what raw material you're using for this bacteria to produce biodiesel. This is our lipid accumulation beta-keto-adipoic pathway, just to show when you use phenolic compound, phenols are broken down and it goes through to make acetyl-CoA, and it goes to lipid synthesis, and lipid synthesis is making your C16, C18 as the main methyl ester biodiesel. So just to show proof of concept, we could take all the biomass material, lignin, cellulose, hemicellulose, and produce useful product out of it. So we can use cellulose and hemicellulose to make ethanol, and the lignin polyphenolic compound could be used to make microbial lipid using rhodococcus bacteria. So we used a lot of different model phenol. We also used lignin. The result is the same. It's producing mostly C16, C18. We tried a lot of different phenolic, vanillic acid, hydrobenzoic acid. They do the same thing. There is no difference. And then we use raw lignin. Raw lignin also work okay so far. So just to show you more results from different raw material, what's happening in cell dry weight, what's happening in personal lipid production. Whatever raw material you use, the bacteria is able to use, produce lipids. And again, just to show you graphically what kind of raw material we used, okay? So in summary, I want to say that from biomass, we could use every component of the biomass. Sugar to make ethanol, phenol to make microbial lipid. This is only proof of concept. We did it in the lab. Need to be scaled up. And this work was done by my former graduate student, Sarah Shield-Menard, for her PhD work at Mississippi State University. So I stole her slides to show you what could be done in lignin, which is the main waste product in lignocellulosic ethanol production. So just to summarize what we talked about, you can make fuel grade ethanol from energy cane that I showed you in which is one of the major crop in Louisiana. We reduced the cost of processing by reducing sulfuric acid use from 3% sulfuric acid pretreatment to 1% sulfuric acid by adding fungus. The idea here is when you harvest sugarcane, you store them. During storage, you use the fungi. So the pretreatment can be reduced using this technology. And then using rhodococcus, you can take care of the phenolic compound in your biomass. Okay. Again, this work was, the phenolic work was done by Sarah Schill, my former student for her PhD work. Just to show you, when you put them in these bags, this sugar cane, this is what that white cereoporiopsis and phenoric look like. So this is exactly trying to mimic during storage, when you store this crop before you process them. So you use less pretreatment material, okay? And this work was done by Dr. Suhadi in my lab, and she was a Fulbright fellow here, okay? I'll stop here, I can take questions, Chandra.
Okay, okay, thank you, Raj.
I might have a lot of time left here. 8:22.
Yeah. So I think Guntur, probably
yes.
You can probably Raj, you can
Stop sharing, yeah?
Yeah, stop sharing. So
yeah.
Could you
is hemicellulose processed by cellulose and cellulose enzyme? No, it is processed by xylosic enzyme. It's totally different class of enzyme. Is protein separation needed? Pretreatment? No, there is no protein in this biomass. It's mainly all lignin, cellulose, hemicellulose, and some pectin. I don't think there's protein separation needed for this. Next question regarding acid hydrolysis for cellulose substrate.
Substrate, yeah,
substrate. What is the best method to neutralize acid for microbial growth? Is there a certain post-treatment after acid hydrolysis of cellulose? Yes, you are. When you pre-treat, after you pre-treat it, you need to do the pH correction for sure, because when you use weak acid, it is okay. But when you use strong acid, yes, you need to correct pH. Yeah, neutralize for microbial use for sure. For lacase, which are better for substrate hydrolysis? Do the enzyme from bacteria more effective than fungus? I think fungal enzymes are better in my opinion, but I never worked with bacterial enzyme for this experiment. So from the literature, it shows that fungal enzymes are better, but I am not sure about bacterial enzyme for, working on this lignin removal. So most of the literature state fungal enzyme work better. Chandra, FX process needs ammonia. It will be difficult for small community regional processing process, either for ammonia handling and ammonia supply. To use biological process pretreatments are relatively a slow process. Yeah, so that slide I showed you is a proposed Michigan State University tech transfer slide. And this is what the Michigan State is proposing in the US for rural economy, mainly because it's patent technology. What they do is they do turnkey project. They come and set up the whole system for you. Of course, there is cost involved, so the investment is needed. But I don't think it will work in poor countries. But this is what the Michigan State, because the FX technology is mainly coming from Michigan State University. I agree with you. So it's a slow process and a relatively slow process for pretreatment. That's why the R&D is needed to improve this technology. As I said, that if a lot of people work on a lot of different countries, definitely you can come up with solution and you can, economically viable system you can work on. So
you mentioned that with fungi, you mentioned that with your fungi, you can reduce the acid until on 1%, right?
That's right, 3 to 1%. Yeah.
From 3 to 1%.
Yeah, from 3% to 1%. Yeah.
So
yeah, when you use fungi. Yes. Yeah.
Yeah. Okay.
Ibnu, how to remove any inhibitor as explained before to produce biodiesel and increase microbial growth without resistance. I took the information about ricin and curtin from biodiesel from plant, and I never know the whole process of how they remove the toxicity of it. But in US, they ban because of production of ricin from castor. Yeah, of course, I think the Jatropha, maybe Chandra may answer that. I don't know how they remove the toxin from biodiesel production from plant. So yeah, I took it from the literature. I don't have my personal experience. I'm just showing you why microbial lipid is better than lipid from plant, because we don't produce this toxicity, toxic compound in microbial lipid production compared to plant processing like Jatropha or castor seeds. That's why I put that slide. But I don't have an answer for how they remove the toxicity. Yeah. All right, any other questions?
So far, there is not yet any new question.
Okay. Probably I went too fast. No, last time I had so many slides, so I went too fast. Of course, I can share the slides with you guys if anybody needs some. So.
Okay. Things I have some problem with my headset. Raj, regarding microbial lipids.
Yeah.
So that one is an intracellular product, that was intracellular, the lipids.
Yeah, you need to break open the cell to release the lipids. Yeah.
And then you have to, how is the downstream processing for the lipid extraction?
It's a sonication or microbial. I mean, that still we are working on it. But personally, I'm working with a chem engineer to deal with that technology. So, but in the lab, what we did a sonication to break open the cell membrane to release the oil. It separates very good once you break the cell membrane. You can easily, all you need is pipe it out, it comes out. So.
Yeah. So that one is the lipid content, I think it's relatively high, right? So
depending on how you grow them, if highly nitrogen limiting condition, you can have high lipid content in this bacteria. So, yeah, we can grow up to 50% by weight. Yeah.
Yep. In your experiments, that one is used the synthetic one, right? So that one.
The slide I showed you are all model compound, phenolic model compound. Yeah. But later on, the student used the lignin as well but I don't have that slide. After she left, she did some postdoc and she did the research. So.
Okay.
Yeah.
So is there any other? So from the hydrolysis, from the hydrolysis, the real one.
Yeah.
It's the real hydrolysis. Is there many studies on the microbial lipids?
No. Only few people are working on this field, because what they do, lignin, they're going to use it as cogen product to burn it in the industry as a fuel source, make electricity. So not many people are working on microbial lipid aspect. Our idea is to show that every part of the plant could be used to make energy. So that's the idea here. So
yeah. So I think there's another question, right, Guntur?
Okay, this one is in English. I can read it. Yeah. As I knew that lignin is not easy to degrade, is there some method to solve the problem in pretreatment? Maybe.
Yeah.
Yeah, lignin, that's why we are trying to separate and get the lignin out so that you can get the cellulose, hemicellulose out. So that technology is there. The other way people are dealing with is genetic engineering. People are producing plants that has less lignin. And people have already done that. They tinker with plant genes. So these plants have very, very less lignin and a lot of cellulose and hemicellulose. So I don't know how far that's going to take before it be commercially viable. But lignin separation is not a problem. Pretreatment works in this case. Okay.
Yeah, I think related with the next question from Josephine. I think which one is using acid or alkaline for the pretreatment process?
Both have some advantages and disadvantages. Acid produces, you have this inhibitor. You also have some sugar release also during acid treatment. You release some sugar. Alkaline treatment, also work is caustic and the pH adjustment after you do that. So each pretreatment has its ups and downs. It depends on the people that work with. Some people prefer acid treatment than alkaline treatment. I prefer acid treatment because of my use of fungus and combining acid treatment together. Okay. So yeah, every technology has its own advantage and disadvantage. Pre-treatment method. Pena, lignin can be converted to various valuable chemicals other than liquid. Have you tried this using microbial route? Yeah, we are thinking of producing a lot of pigments. So cosmetic industries. So you can produce different pigments. That's one idea we are looking at. This was the first proof of concept to show it could be produced microbial lipids. Yeah, you can produce other products. One of the major one is a lot of different kinds of pigments may be produced using different organisms, different bacteria. Yeah. Yeah, we used a variety of consortium of enzymes. I showed you in one slide what are the different enzymes we used, okay? I went too fast, but I used 5 different cocktail of enzyme because we wanted to produce the maximum theoretical yield. So yeah, it was not single enzyme, it's a multiple cocktail of enzyme. Yeah. I can share that slide with you. You can check that slide for what are the different enzymes we used, yeah.
It's a different, okay.
You said that the microbes produce microlipid grown in nitrogen limiting condition. Yeah. So the idea here is when bacteria grow in ideal growth conditions, they have a lot of nutrient available. They don't have to make fat reserve, right? So what they're doing here is when you have plenty of carbon and less nitrogen available, the bacteria is making storage of food. They're converting this carbon to lipids. So they can use them as future use to grow. That's a purpose in nature, right? So that's the reason, that's the one we are exploiting. So you grow these bacteria in nitrogen limiting condition. So we turn the bacterial gene on to make more lipids. So that's why we do this. Yeah. Because in nature, that's what the bacteria does. when it has nitrogen limiting condition, when there is more carbon available, instead of growing, producing more cells, what they do, they make more lipids for future use. It's like a survival mechanism for bacteria. And that's exactly what we are exploiting.
Yeah. Also, a lipid doesn't contain any nitrogen, right? So that one is in the structure of the.
There is no. It's all carbon hydrogen. Yeah.
Yeah. Just only.
Yeah, the excess carbon available to the bacteria is putting them all together as a lipid. So no nitrogen in it. Yeah.
Okay. So for the, is there any others? Probably from the Zoom, if you have a question, you may address directly to Professor Raj. If you are willing to ask question. And is there any other question from you? You may.
Prof. Chandra.
Oh yeah, please, Willie.
Yeah. Maybe in future can make another sources from the cassava.
Cassava, it's 100% starch, right? You shouldn't have any problem.
Yeah, because last time some of our friend is making bioethanol from the cassava, and the production also quite high.
Yeah. So cassava is included in the first generation, right?
That's right. It's first generation. Yeah. Like a corn and sugar ethanol. Yeah, that technology is mature technology, because all you need is amylase to make your sugar, and then you get the fermentation process.
I think Sandy, you raise your hand, please. Sandy? Oh, no, he raised his hand, but
is there a question there? Someone has a question. I can't see it. Can you read it?
Yeah. Is there any microorganism besides yeast to produce ethanol applied in the industry?
Mostly yeast, because that technology is really commercialized. So there are a lot of companies that sell yeast, but the organism I'm working on is E. coli, but a lot of people are working on different organisms like Zymomonas, Clostridium to make butanol. So there are a lot of biocatalysts you can work on, but the commercially produced ethanol from corn, and also some of those plant I showed you that make ethanol from cellulosic material are using yeast. Some of them are using yeast that are recombinant yeast, because they have to process the xylosic sugar as well.
Yes. So that in the US, I think now is with the Joe Biden's government seems to be a promising now, right?
Yeah. He's now favoring green technology. That's his policies are now improving to biofuels and green technology. He's also stopped some of those petroleum drilling in federal land. Before Trump administration, he opened up federal land for oil drilling, but now he put a moratorium to stop that. So we are hoping this green technology will take up fast again in the US.
So, and also you are mentioning about you are straight in your presentation about the liquid fuel. So that how is the electricity car will replace the petrol car? when will it be?
It's a long way to go. But if you look at some of those car companies, they are already announcing they're going to stop producing petrol run car by 2030, 2035. Some of those companies. So they are making big plans to switch to electric cars. So yeah, I hope it works. But yeah, yeah.
Okay. Is still any other question from you? Probably you may raise your hand if you are in the Zoom. If you still have, probably because Avalina is working on the microalgae. Probably Avalina, you have you like to raise the question regarding the algae's possibility to produce fuel. Is now how is the fuel from the microalgae in US research?
We are lagging behind in algal technology. Some companies started up when the oil price went down, everybody stopped. When oil price really went down, it's not viable to make microalgae lipids. So, but the South Korea is leading in this field. So there's a lot of labs doing this work in South Korea. So. In the US, we are lagging behind in microalgae technology. So.
Yes. Because one of my students graduated from the South Korean universities and that's working on the fuel from the microalgae.
Microalgae,
yeah.
KAIST is the institute.
Yeah. Oh, KAIST is the leading, right?
Yeah. In this area. Yeah.
Please, if you have any other question, or otherwise, probably we may close our session.
Okay.
Gunter, maybe we would like to close our lecture today, and thank you to Professor Ramaraj Boopathy. You are already in the last 4 weeks to give the guest lecture to us. And then hopefully it will give you new knowledge to you, to the student, and also not only student, but I saw that my colleagues from other universities and other institutions here, and also all the lecture note or lecture handout will be in the Google Drive, and I think Guntur will share the link, so you may download from the Google Drive. So I think so, Guntur, you may close the in the YouTube. So that we like to